we can proceed perfectly well. And I'd like to begin with a brief introduction about Center for Predictive Human Model Systems, the work that we do, and then we will briefly move on to Dr. Janani's talk. So Center for Predictive Human Model Systems, or CPHMS as we call it, is a science and policy think tank that advocates for human relevant research in India. And this is to do with biomedical research as well as drug development. So you may be aware that the pills that we consume, such as paracetamol or a painkiller of any sort, they usually take a long time to come to us. This is easily in terms of 10 years or so. And the reason that it takes so long for them to come to us is because they undergo a rigorous process of validation and research. This process involves laboratory formulation, testing in various capacities, and as of now, testing models are mostly animals, then human clinical testing, and finally, it is approved by regulatory authorities and then comes to the market. Now, one of the reasons why this process is inefficient is because animals are used before we test them on humans. Now, while it sounds very convincing that animals could be the first line of testing before humans, it's not necessarily perfect because animals do not predict human responses as well as we think they would. Even between animals, there's a lot of variation as we look at what responses are generated. So clearly, we need better models to predict human responses. And these models are what are predicted towards the right. So these are modern models called organoids, organs on chip, computer generated predictions, which are really helping us come closer to what human responses look like. So CPHMS is an organization that promotes work with these models. And what we've seen is in the West, these models are increasingly adopted over the past 10 years. But in India, it's not working quite as fast as we'd like it to. And so our effort is to raise awareness, to promote education about these systems, and also to make sure that there are regulations in place which make sure that these models can be adopted. So we interact with various people, such as investors, industry, common man, scientists, students like you, regulators, so as to make sure that the entire ecosystem is friendly enough to the adoption of these technologies. As you're here today, I'm very happy to say that this is the 25th webinar, and we've had many webinars in the past focusing on computation or on these models. You can access them on our QR codes here, and we hope that your enthusiasm continues so that we can have many more of these. Another interesting series that we've been promoting throughout the year is called Back to the Future. Oftentimes, students hear about these models, but they're not necessarily aware of what exactly goes into these models, how exactly are experiments conducted. They just remain big names in the textbook. And so what we've done, our team has done, is that they've gone to such labs, filmed experiments with these lab members who work with them on a regular basis. And so we have them on a YouTube channel. So feel free to scan the QR code, go there and watch all the exciting episodes. They're in a very friendly manner. So you don't necessarily need any prior information at all. The first episode focused on organs on chip, and it was wound infection on a chip. The second one was on mini organs. The third was on computational biology. Our most recent one on advanced imaging like fMRI or TMS, techniques which help you to visualize what's happening in a human body, is also out. And so you can also feel free to watch that. Some other things that we do, we have a quarterly newsletter informing what's the most recent thing that's happening in the field right now. We publish academic papers as well as white papers, which talk about this field and how it's moving forward, what are the challenges, and what can be done to really improve the stature of this field in general. Another thing, another activity at CPHMS is having education and training. As I was mentioning, the field isn't so advanced in India as it is outside. So several areas need a push, and some of these are understanding what are adverse outcome pathways, how microphysiological systems work, what they are, and newer technologies like 3D bioprinting. So we've had workshops on all these, and I'm very happy to say that Dr. Janani is going to talk to us on adverse outcome pathways, which has been something that we have been very closely fond of, and we hope to promote more largely in future. Some of our initiatives also include giving grants to people. Again, we've worked on AOPs in the past. We have received a grant to work on understanding the landscape of economics of these models like organoids, organs on chips in India. And we also talk to policymakers. In the sense, we've had over 3 roundtable conferences with several authorities to push the field forward in India. Lastly, what I'd like to talk to you about is a database that we've created. So you must be wondering, who are the scientists in India who are working in this area, and how can we access them? So CPHM has created a database wherein you can look at the map of India and identify scientists all over the country working in this field, what exactly it is they work on, how can you contact them, and get in touch with them for internships, fellowships, collaborations. So we hope that it will be useful for you as well. We've received tremendous response. Do scan the code here. You can access it here. And if you are a scientist working in this field, you can also scan this QR code and join the community. So with this, I'd like to thank our team, Dr. Madhu, who is our CEO and who has been at the head of it. It has been his vision, really. Surat, who was in my position earlier, and she's now a consultant advising us on how we could move forward. Anushka and Gautami for helping me every day in everything that we do. With this, I'd like to end my introduction on CPHMS, and we can now move on to Dr. Janani's talk. Very briefly, I'd like to introduce Janani. So it is my pleasure to have her on our webinar today. At present, Janani is a research data steward at Novo Nordisk in Bangalore. She graduated with a PhD from the Institute of Mathematical Sciences, Chennai, where she worked with Dr. Arijit Samal on data visualization, programming, database design, systems biology. For her excellent work during her thesis, she was awarded the Outstanding Doctoral Student Award by Homi Bhabha National Institute in 2023. Today, she's going to elaborate on one of her PhD thesis chapters, which dealt with adverse outcome pathways. AOPs are what help us predict the impact of certain chemicals like cement, harmful cosmetic ingredients, pesticides, etc. And these are ingredients that we encounter on a daily basis. It's very important to know what they are and how they cause disruption in our body. On a personal note, it is very lovely to have a young and ambitious scientist like Janani with us today, and I'm sure she's going to inspire us all. So looking forward to your talk, Janani. Over to you.
Thanks, Kasturi. Thanks for the nice introduction. I'll just share my screen.
Sure.
I guess I can't share my screen. It says host disable participant screen sharing.
Try now.
Yeah.
Yeah, we can see your screen.
Okay, great. So hi everyone. So today I'll be presenting one of the topic which is very interesting, and this is very new topic to everyone also, which is adverse outcome pathways. And this is a part of this was actually not planned while doing my PhD. So we were actually focusing on a lot of environmental chemicals which were of concern to human health. And suddenly when we explored this one and to understand more of endocrine disruption mechanism. So maybe let me start, like how did we start this? So initially our focus was more of the environmental chemicals. So in our day-to-day life, we are being exposed to a lot of chemicals in the environment, starting from water that we drink and air pollution, cosmetics, medicines, etc. So some of these chemicals can disrupt our endocrine system, leading to different diseases like related to development, neurology, metabolism, uh, and also reproduction. And these chemicals are called as endocrine disrupting chemicals. So these endocrine disrupting chemicals it can be anywhere in the environment, and we wanted to first work on this endocrine disrupting chemicals. So coming to endocrine system, so our body has a lot of glands that can secrete hormone, and these hormones are responsible for various functioning of our body. So it can be growth, development, metabolism, reproduction. So, and also these are responsible, the hormones are responsible for the normal functioning of human beings. And the endocrine system, EDCs always, what happens is it tries to mimic the natural hormones, and it doesn't allow the hormones to bind to its receptor, leading to some adverse cellular responses. So that's how it leads to different adverse outcomes and disorders. So first, what we wanted to do is we wanted to identify the EDCs which can cause the endocrine disruption. And when we were looking for EDCs, we didn't find any right description or like definition or any validated assays which can be used to identify EDCs. So what we decided is we built a 4-stage workflow where we can curate the information which are in existing articles or reports to find the EDCs with endocrine disrupting chemicals. To give an overview, so in the first stage, we undergone literature mining where we have taken all the literature and existing reports from some of the existing databases. And then we have looked for specific keywords which are related to EDCs in those literature. And then we came up with 14,297 potential studies which are on EDCs. Then further, we did some literature filter based on the study type and test organism, because we are more interested to look what's happening in human other than other animals. And many articles and many studies are related to monkeys and other animals for the endocrine disruption. So we wanted to focus on human and rodents. So we just filtered all the studies only to focus on humans or rodents. And further, what we did is we collected all the tested chemicals from those papers, from the research articles. So, someone have any doubt?
Oh, please go ahead, Janani.
Yeah, okay. So, so then we have just compiled all the chemicals, tested chemicals from these papers. Then so this resulted in 1,626 chemicals, potential EDCs. And then we were just checking for all these chemicals if they have any associated endocrine mediated effects or not. So if the paper says that the chemical has led to some potential endocrine disruption effect, so we take the chemical as EDC, and also we compile what effect it has been observed, and also we are collecting at what dosage the EDC has created the effect, created the effect. So we have totally 686 chemicals which were identified as EDCs, and these were from 1,796 published literature. And we have, as I said, we have, apart from these EDCs, we also have the dosage information and also the endocrine mediated adverse effects which are associated with these chemicals. And further, the effects, whatever effects we have found, we have unified them into 514 effects so that we'll not have any duplicates. So we have unified them, and also we have further categorized them into different systems at which we observe. So if you're interested to look more about this, then you can go into a database called DIDIC, which is database of endocrine disrupting chemicals and their toxicity profiles. So this has all the information which we have collected from different articles. So as we have compiled all the endpoints, the adverse effects, so we have further classified based on the different systems where we observe. For example, atrophy, embryonic development. So if the chemical affects the embryonic development, then the endpoint is categorized into developmental system. And similarly, there are some cancer-related effects, reproduction, hepatic, metabolic, neurological, immunological, etc. And when we analyzed based on our data, we find that most of the EDCs are likely to affect reproductive system than anything else. And also what we observed is all these EDCs are, some of the EDCs are creating like disrupting most of the systems. So maybe one EDC can affect 7 systems also. So in such cases, we want to understand how this is happening, and we wanted to understand at the systems level, and how we want to understand also the mechanism, how this is happening. So for that, we thought we can link the effects to adverse outcome pathways. And so we have explored this topic a bit. So what are adverse outcome pathways? So here you can see a list of responses. So when a chemical is exposed, then it initially triggers a molecular response at receptor level. Then it goes to the organelle level response, cellular, tissue, organ, and organism level response. So this is how it affects different levels of biological organization. So, and the AOP covers all these systems. So it covers events at all the systems. So here, these molecular interaction is called as molecular initiating event, and all other events or responses are called as key events. And these were related by key event relationship. And the final organism level response will be called as AO. And the whole of this chain will be called as adverse outcome pathway. So like after this, I'll be just calling these key events as KEs, adverse outcome as AOs, and also molecular initiating as MIEs. So I'll just tell you how we have leveraged this AOP concept to study the endocrine disruption mechanism. So this is the AOP which I mentioned, which is in a simple linear representation form. Consider these two are AOPs, and they have common KEs, the key events. And these can be combined together as one AOP network by combining this using this shared key event. So this similarly, we can just create a whole AOP network. And also further, we can just derive a network for a specific endocrine. For example, we want to study something related to endocrine system. So we can just pick the key events which are related to endocrine system and create a derived AOP network for endocrine disruption. So this is the basic of how we can create a network with some of the linear AOPs. So we have created a workflow where we can derive the AOP which is specific to endocrine system, and also finally also the analysis of the endocrine AOP network. So I'll just go one by one on it. So first is we want to compile and curate the data from AOP Wiki. So AOP Wiki is a existing repository which has all the data about AOPs, key events, key event relationship, and also the stressors. So here the stressors could be the chemical stressors and also the non-chemical stressors like radiation, microorganisms, etc. So when you I'll just give a glimpse of how AOP Wiki works, because this is the data source we are picking to study the AOP network for endocrine disruption. So just when you click on this AOP, so you will get list of AOPs which is there. So here it says 452 AOPs are there, and when you click on this, so it gives a list of information about the specific AOP called DNA damage leading to a mutation breast cancer. And also there are a list of things which you can get from this page. So it will have what are events, and also the relationship between key events. Also, it will have the stressors, and it will also give some life stage information. For example, the event happens at development level, or the event happens in particular age or something, life stage or some adolescence or something like that. And you will also have the taxonomic information where you will where the data has come from, like which organism the data has come from, and also the sex applicability. So breast cancer could be specific to female, so such information will be there. Then once you go into events, in this page, you can have a list of events here. So these are the MIEs. So initially it starts with the molecular initiating event, which is increased damage and mutation. Then there are list of KEs, key events, and then finally the adverse outcome is the breast cancer. So these this molecular initiating event will be mostly at the molecular level, and the adverse outcome will be at the organism level. And these are the other different level response, like the level of responses at cellular and organelle or tissue. So we have a download page in AOP Wiki where you can you can scrape the data, or there are XML which you can import and then you can work on it. So finally, based on that, we have extracted these information from the AOP Wiki. So we have 316 AOPs when we have extracted. This was done by end of 2021. So now there are a lot more updates. And we had KEs, KERs, and stressors. And once we extract, we want to find the complete information. Because this AOP Wiki is being updated regularly by scientists, so we may not have complete information, but we want to work on the AOPs which have the complete information. So we did some list of filtering here. So we removed some archived AOPs which are no longer under use, and we removed the empty AOPs. So this AOP will be just have the number, and they will not have any key events or any KERs. And also we also remove the AOPs which do not have MIE and also the AO information. And also we try to find the weakly connected components, and we remove the AOPs which has more than one weakly connected component. Then we try to find path from MIE to AO, MIE to KE, and KE to AO, and within each of AOP. So if there is a definite path exists between these, then we take those AOP. So finally, we get the confident set of AOPs, which is curated. And finally, from 316 AOPs, this resulted in 161, and with 635 KEs and 810 KERs. Then secondly, since we want to derive the information only specific to endocrine system, so we have taken this confidence set, and then we try to filter the key events based on these keywords. So if these key events has any of these keywords related to endocrine glands, hormones, receptors, disorders, and also endpoints, then we consider them as endocrine specific key events. So finally, we got a list of 294 key events, which is across 151 AOPs and 810 KERs. And also, once we filter it, filter based on endocrine specific KEs, we have to make sure that it's, again, we have a complete information. So we just finally selected only the AOPs which has at least one MIE and one AO. So this resulted in 48 AOPs finally. And this was taken forward to do other set of analysis or to construct the endocrine AOP network. Then it comes to endocrine AOP network construction. So as I said before, so here are two AOPs, and if they have a common key event, then it can be assembled together to form a single AOP network. So in such way, based on the shared key events among the 48 ED AOPs which we have extracted before, we have constructed a ED AOP network. And this is the ED AOP network with 48 AOPs. Then we perform some graph theoretic analysis for this ED AOP network. And we find that there are two largest connected components. And totally there are 7 connected components, but these two are the largest connected components. So we just took these two to study more about the endocrine disruption so that it can give some insights on what is happening at the level of cellular or organ or tissue, etc. So maybe here in this presentation, I'll just go with explaining about one largest connected components, which is C1. So both of these largest connected components has 12 ED-AOPs. So here is the expanded version of the largest connected components, C1. So here the color coding represent different components of the AOPs. So here the yellow ones represent the molecular initiating event. The green ones represent the key events, and the red ones represent the adverse outcomes. And the ones which are bordered by outlined by red, these are the shared key events among different two or more AOPs. And then what we did is we have applied some network measures for these connected components. So what I did was we have used the cytoscape to calculate some of these properties, which is in degree, out degree, betweenness, and eccentricity. So in degree will tell you how many number of incoming KEs. For example, for this one, there are 3 in degrees. So 3 of them converge here and 3 in degrees. And similarly, out degree is outgoing one. So here there are again 4 out degrees here. So this will be important to study. Maybe I'll just get how this will be important in the next slide. And also then we have calculated the betweenness and also the eccentricity. So betweenness measure will give an idea of how, what are the key events which are crucial for this biological perturbation, which is then eccentricity is, it will give an idea of how far the key event is from. So if the eccentricity score is really high, then it's like it's very far. And if it is very less, then it is the centrally located key events. So this will give an idea of how important each key events are. Then once we study that, we also try to just classify all these key events into different systems to know how these systems are connected. Because in general, endocrine, all the endocrine disorders tend to comorbid with each other. For example, people with diabetes might be linked to some of the other metabolic disorders like obesity, or also even sometimes the cardiac disorders. So these are always comorbiding with each other. So we want to understand if we can get any of the insights on how they are connected at systems level. So here we have these key events. These events are colored based on the systems. So here the pink ones are the reproductive systems. So for example, impaired fertility. So this was classified as reproduction. And here the something related to cell proliferation of hepatocytes. And this is again classified as hepatic. And also here the green ones, these are metabolic, which are related to like thyroid or some metabolic functions. And these orange ones are related to the neurological, like the cognitive function, etc. So we want to see how these are interconnected. So for this, we have used the measures which we have computed before. So we found the point of convergence where the in degrees are more than the out degrees. So these, for example, this has more number of point of convergence, and also this particular AO, adverse outcome, has more point of convergence. And so this can be further used to see how additive effects can be found. So if, for example, any of the EDCs or any of the stressors can bind to this receptor, this is an MIE. So like here there is androgen receptor and a CAR receptor. So if any of them bind to this, then it will finally lead to this specific function as an additive effect. So such information can be pursued from these point of convergence. And also it can finally lead to some hepatic issues like hepatocellular adenomas and carcinomas. And then we have a point of divergence here. So here you can see this is the activation of PPAR gamma, and here this is diverged into two different key events which are specific to reproduction. So here we have an idea how this hepatic things are just diverged into the reproduction system. So this will have a better idea, and also this also suggests some endpoints and assays which are which can be used further for the chemical risk assessment. So maybe we can plan some assays over this PPAR gamma, which can give some insights of what will happen next. So if this is getting activated, then it may lead to some of the downstream effects related to something reproduction or even the hepatocellular carcinoma. So these are some of the insights which you can get from point of convergence and divergence. And also we'll get some of the emergent paths. So here emergent paths are here the emergent paths are the new path which we will find after we construct the AOP network. For example, this consider this as one AOP network, and there is some other network also which can be formed like this. So this is a emergent path. So this will give clear idea of predicting the unknown adverse effects. So this will give, maybe if we just know that this will cause a particular thing, maybe it will also lead to some other effects also. So these information can we get from emergent path. And also we can link the we have also linked the KEs to different chemical stressors, especially the EDCs which we compiled previously. And we find that there are 16 EDCs which are associated with this component. So that's it. So finally, this network, it gives a landscape of potential adverse outcomes which are associated with the EDC's exposure. And also the graph theoretic approach gave some mechanistic insights which can be even explored more based on your interests. And the network can be used as a tool for risk assessment. And also we can derive this AOP for some other interest also. Maybe as Kasturi was mentioning, if we want to learn something for the cancer, then we can derive it specific to the cancer. But however, one limitation would be we are dependent on the data from AOP Wiki. And also the AOP Wiki is actively updated, and also it is it is very live. So everybody are keep working on it. So the information keep changing, and we are also very much limited by the existing knowledge. So these are some of the publications which are related to endocrine disrupting chemicals, and which you can go through. And these are some of the other environmental chemicals which I have focused during my PhD. You can just have a look at it if you are interested on any of these. And finally, I would like to thank my thesis advisor, Arijit, for all the support, and also co-authors and IMSC. And also I thank Kasturi for inviting me for this talk. So any questions?
Thanks, Janani. It was such a wonderful talk.
Thank you.
If any of the participants have questions, you can unmute yourself and ask the questions yourself. Else you can also put them in the chat box and I'll be happy to ask them for you. So before we take questions from the participants, maybe I could begin with a question from my side. I was wondering, in the case of AOP related information, is it possible to identify what event occurs first, which leads to the next event, and what are the implications of these in the big scenario?
Yeah, Kasturi, we can. It is based on, those information are available in AOP wiki already. So we can go into that and we can check that. So maybe I'm not sure if I can share the AOP wiki page. But yeah, all these information which you have asked, you can get it from AOP wiki, but it's very much limited to what information it's already there.
I see. And so from these studies that you have done, is it possible to draw certain conclusions as to how exactly the toxin acts on the human body and
yes,
what can be done? Okay,
yes.
I see. Do you have certain examples of things that could be done, some greater learnings which are evident from your studies?
Actually I don't have any specific figure related to this. So we have some 16 endocrine disrupting chemicals which are associated with different key events here. So for example, consider some X chemical is associated with this specific key event. Then we can definitely predict what are the possible adverse effects. So it could be either the hepatocellular carcinoma here, or it could be either it can lead to something related to metabolism, or reproduction, or something related to even the metabolism. So we know how the connections are based on this network.
Right. So in terms of learning about AOPs, what kind of background do you need to work with these? Like what kind of educational background, what internships should you have done?
For me, I think it's biology is, of course, I think you need it. And also equally, you need some idea about networks. If you are going to apply something related to networks, then it's better if you have some background about network measures and network biology.
Okay, great. Thanks. So Debankshi is asking a question. How can we use AOPs to study carcinogenic pollutants for anti-cancer drug development?
Okay, so as I was mentioning, so here we see that the point of divergence, it can suggest a novel endpoints or assays for better chemical risk assessment. So if this is a point of divergence, for example, so we can design some assays over this, and we can see if the chemical is binding to this, then we'll be able to find if we can what adverse outcome it can lead to. but I'm not sure if you can find some anti-cancerous effect, but I think it's just limited to what adverse outcomes it can lead to.
Right. Deepthi has a question. Which tool have you used for pathway reconstruction or network development? Whether any flux balance analysis employed here?
No, no flux balance. It's just topological measures. This was computed using network analyzer app in Cytoscape.
Okay. So I think these are the only 2 or 3 questions. Thank you so much, Janani, for your time. And
Thank you, Kasturi.
if any participants are interested in connecting with her over any questions, please feel free to reach out to her on her LinkedIn profile.
Yeah.
Yeah, it was great to have you all, and see you in our next webinar.
Yeah. Thank you, everyone. Yeah.
Bye.